So hello and good morning everyone. So today we are from Group 1 and we're gonna presenting about the key process of gen expression. So the gen expression in the simple way is the process of using a gen information to make a protein. But in a full sentence to understand it, I will say that gen expression is a process which the information encoded in gen is used to synthesize a functional gen product. For example, like protein or functional RNA molecule, which determine a cell structure and a function. So here you guys can see below here there is a diagram. And this diagram is actually what Central Dogma tried to explain to us. So DNA will undergo transcription, which is one of the process for the gene expression to form RNA, and then RNA will undergo translation to form a protein. So meaning that gene expression have two main processes involved, which is transcription and translation.
Okay, so now let's start with transcription. What are the components for protein synthesis and their functions? So first of all, we have DNA strand. Function of DNA strand is to expose the bases of specific segment of one strand of DNA that serve as template to synthesize new mRNA strand, which are also called antisense strand. The other component of protein synthesis is protein coding gene. The protein coding gene codes the synthesis of a specific protein. The examples of protein coding genes are promoter, RNA coding sequence, and terminator. Lastly, another component is RNA polymerase. So the function of RNA polymerase is to uncoil the protein coding gene, or so-called unwinding the DNA strands during initiation process. Besides that, it also adds free RNA nucleotides in the 5' to 3' direction during elongation process. Now, what are the different stages of transcription? There are 3 stages of transcription, which are initiation, elongation, and termination. So firstly, initiation. The transcription is initiated with the unwinding of DNA strands by RNA polymerase. So, in prokaryotes, a specific recognition by RNA polymerase of the promoter base sequence. In eukaryotes, the RNA polymerase recognizes a complex of proteins which help binding of enzymes to promoter sequence. So the next stage is elongation. As elongation is when RNA polymerase move along the 3' to 5' template strand or antisense strand, and free RNA nucleotides are added in the 5' to 3' direction according to the base pairing rules, except that uracil are replaced by thymine to pair with adenine. Okay, lastly, the last stage, which is the third stage, is termination. Okay, so termination is when RNA polymerase encounters the terminator sequence on the DNA template. RNA transcript is released from the DNA template, followed by the RNA polymerase. In prokaryotes, RNA transcript can be immediately translated by ribosomes, whereas in eukaryotes, RNA must go through RNA processing before leaving the nucleus. Yay.
So I'm going to talk about RNA processing and RNA splicing, and this only occurs in eukaryotic cells. In eukaryotic, genes in DNA contains non-coding sequence and also coding sequence, and non-coding sequence are known as introns and coding sequence are known as extrons. So during the transcription process, both introns and extrons will be transcribed into pre-mRNA, and only exons are needed to be translated. So introns are removed through the splicing process, and the exons are joined together to produce this mature mRNA. So in the end, this mature mRNA will leave the nucleus to be translated in the ribosome in the cytoplasm. You could see that both in eukaryotic cell, both transcription and RNA processing occurs in the nucleus, while the translation of mRNA occurs outside the nucleus, which is in the cytoplasm. In contrast, in prokaryotic cell, transcription and translation occurs almost at the same time, which means that as soon as mRNA in the prokaryotic cell is being transcribed, translation of mRNA occurs immediately after that, which means there is no need for prokaryotic cell to undergo RNA processing or RNA splicing.
Now I will be explaining about translation, which is the second main stage of gene expression. What is translation? Translation is the process of converting mRNA sequence into a chain of amino acids that will form a protein. This process happens at the ribosomes, which are found in cytoplasm. There are three main components involved in translation: mRNA, which carries the genetic code from DNA, tRNA, which binds the correct amino acids, and the ribosome, which acts like a factory that assembles the protein. Next, translation takes place in three stages: initiation, elongation, and termination. Let's go through each stage in order. First, during initiation, the small ribosomal subunit binds to the mRNA near the start codon, which is AUG. Then, a tRNA molecule carrying the amino acid methionine with an anticodon UAC pairs with the start codon, which is AUG. After that, the large ribosomal subunit joins to form the complete ribosome. The first tRNA now sits in what we call the P sites of the ribosome. Next comes the elongation stage. During this stage, tRNA molecules carrying specific amino acids enter the A sites of the ribosome. The anticodon of the tRNA pairs with the matching codon on the mRNA. A peptide bond then forms between the amino acid in the P site and the new amino acid in the A site. The ribosome then moves along the mRNA. This movement is called translocation, which shifts the tRNAs from the A site to the P site and then to the E site where they exit. This process repeats, adding one amino acid at a time until a long peptide chain or protein is built.
In translation, termination occurs when a ribosome reaches a stop codon on the mRNA. The stop codons are UAA, UAG, and UGA, and there are no tRNA molecules with complementary anticodons for them. Instead of a tRNA, a protein release factor binds to the A side of the ribosome. The polypeptide chain detaches from the tRNA and begins to fall into its final protein shape. Finally, the RNA ribosome complex disintegrates, releasing the large subunit, small subunit, tRNA, and mRNA. In conclusion, it shows that genetic information flows from DNA to RNA and then to proteins, a process known as the central dogma of molecular biology. DNA stores genetic information which is transcribed into mRNA. Then the mRNA carries this information in the form of codons, which are sequences of 3 bases that code for specific amino acids. During translation, these codons are read to form proteins. These proteins determine the physical traits or characteristics of an organism. Therefore, DNA defines the genotype, while the resulting proteins determine the phenotype, which is the expressed physical appearance or observable trait.